Okay, folks, if you don't mind, we're going to get started. So just a couple of quick administrative notes before I get into the lecture material. So number one, your visual summary marks, some of you have seen them, some of you have not. They will all be posted by tomorrow at 12 noon. We just wanted to make sure everyone's mark is more or less in the same range, and we did a little bit of adjustment to make sure everyone's sort of in the upper range. Not that we're trying to lower marks, we want to actually make sure everyone's doing really well. So those will all be posted for you. There are a lot of 5 out of 5s, and I'm sure that you'll get some comments back as well from your TA. I also marked a few of those as well, so you'll see some of the grades are actually coming from me. And I will be around next week, so next week you can come in during the fall break, after fall break as well, and you can come in and take a look at your term test. It's best to try to email me. I'll be in and out of the office, but I will be around the entire week next week. So please do come in and check out your term tests if you so wish to do so. So I'm going to take you back to what we were talking about in the last lecture. So in the last lecture, we were looking at whether or not all BDNF is good. The consensus is that not all BDNF is good. Some forms of BDNF are better than other forms of BDNF. Even if you are exercising, even if your neurons have the capacity to actually produce BDNF, not all BDNF gets secreted in the same way. And as I mentioned to you in the last lecture, you should be pretty comfortable going back and explaining how you might actually test whether or not BDNF is being released if you are able to transfect that into a cell. We talked about the original Egan paper back in 2003 that looked at different BDNF polymorphisms. That has turned out to be very, very accurate, and now we look at BDNF polymorphisms in a number of different conditions and a number of different diseases. And in fact, in many different conditions, including eating disorders, so for example, in anorexia, people look to see whether or not people who have anorexia also have this polymorphism at position 66 of either a valine or a methionine. There has been some implication that people that have epileptic seizures might have more of one isoform than the other, and people have looked at seizure activity and the correlation with the valine 66 methionine polymorphism as well. All of this has actually led to looking at what is the key role, what is the essential role, especially during development, of a very important factor like BDNF. And it turns out, again, Egan is one of the authors on this paper, that if you were to go and take a look, this variation actually makes a big difference in terms of the cortical morphology. This is not, the next figure is not actually taken from that particular paper. It is sort of the downstream effector of BDNF. And what these individuals did was they wanted to know if BDNF is really all that important. So BDNF, different isoforms of BDNF might affect the cortex and the development of the cortex and the size of the cortex. If it really is that important, we should be able to see some of the downstream effectors. So BDNF binds to its receptor, the TrkB receptor. among others, and then once it activates that, it will activate the RAS pathway activates MAP kinase, etc. So they wanted to actually know, is this true? So if we were to stimulate the other parts of the pathway artificially, can we actually increase the size of the brain? And this was their experiment. So they actually generated a transgenic mouse, and some of the details are important, some of these details are not important. We have previously looked at the essential role of different promoters, and here we're looking at a synapsin 1 promoter. So any cell that has synapsin 1 will be able to express this particular molecule known as RAS. And we've looked at RAS previously, and RAS is one of those factors that is involved in the downstream cascade following BDNF binding. So what this is suggesting is that the promoter, which is expressed only in neurons, the promoter is not expressed in neurons. The promoter will allow the expression only in neurons of the construct that immediately follows after it. So synapsin 1 is specific for neurons, and only neurons will be able to drive the expression of RAS. So glial cells don't have the capacity to drive RAS. Oligodendrocytes, they don't have the capacity to drive RAS. Only neurons that contain synapsin, and all neurons contain synapsin, will be able to overexpress the RAS protein. Instead of using GFP, they used a marker known as LAC-Z, and in the presence of a substrate known as XGAL, when you were to stain the brain, you would actually be able to see this blue staining. Normally we wouldn't see it, and the LAC-Z is like GFP. It's a marker that indeed the cells have actually expressed RAS as well as LAC-Z. And you can see we get lots and lots of blue staining here, which suggests that neurons have actually overexpressed RAS. So what you're looking at here on the left is a fusion. We're looking at two separate brains that are age matched. So they're exactly the same age. One of the brains is a wild type animal, and we have actually fused it. We've cut it in half, and we've actually fused it with a same age-matched animal that has overexpressed RAS using this particular construct. So again, this is all downstream of BDNF. If BDNF does its job, in fact, we should actually see the effects of having overexpression of BDNF, or the downstream effector of BDNF, in this case RAS. And indeed, what we see is the brain itself actually physically gets bigger. So the brain, if you were to take a look from the olfactory bulb all the way down to the cerebellum, this is the normal length, and you can see that indeed the brain is both longer. And if you look at the cerebellum is bigger. And if you were to compare the cortex is bigger. The hippocampus is certainly bigger. And it suggests that all of the different structures, if we overexpress the downstream effector of BDNF, does indeed lead to a bigger brain. So is BDNF essential? Yes. Is this pathway of RAS activation essential? It is indeed also essential, and you will develop a bigger brain, as you see here on the right. So again, just as a review for you, one of the three pathways is about cell growth. We've just looked at the pathway that's involved in cell growth. We also know that neurotrophic factors will also engage in two other aspects of signaling. So these growth factors will activate RAS, and I've just shown you the RAS pathway. And this RAS pathway will also affect survival. It will inhibit apoptosis. And in fact, it will also inhibit the apoptotic pathway directly. So not only is it enhancing cell survival, having BDNF, for example, will also inhibit apoptosis. And I talked to you very briefly in the last lecture about this phenomenon that neurons actually look like they are programmed to die. In fact, they're very fragile. If you were to damage neurons in your brain, they're almost set to undergo this programmed cell death or apoptosis. So whether it's through neurotrophin deficiency, we don't have enough neurotrophic factors, we would activate this pathway, activate the Janus kinase, and will also activate a number of other pro-apoptotic factors leading to cell death. This also will create a smaller brain, so the neurotrophic factors will enhance cell survival and will also enhance cell growth. And the last pathway that I talked to you about very briefly was the phospholipase C pathway, which isn't in any of these diagrams here. So that was sort of a review. Do you have any questions on any of that material or anything that I can clear up for you now? Yes? So the ventricle here, like the So that's a really good question. One of the reasons why it's bigger is all the structures actually get bigger within this particular brain. So this is actually not a good sort of overexpression because the brain actually doesn't have anywhere to grow. The bones don't get any bigger. The cranial capacity doesn't get any bigger in this animal either. So this is somewhat artifactual. It's a good question. So if you don't have any other questions, I'm going to take you through this technology. I don't need you to know all the details of this technology. I'm going to point out a couple of things to you. Right now, if you were to take a look at modern neuroscience is at a really critical junction. So in the 16th century in astronomy, people were building these really big, powerful telescopes. They were trying to look farther and farther out into space, and it was a race to see who could build the biggest telescopes to see the farthest in our solar system and farthest within in terms of different celestial bodies. And this is sort of where we're at right now. We're sort of in the 16th century in terms of what we're trying to do. We need to be able to see these molecules. We need to be able to see BDNF. We need to be able to see cell death. It's really hard for us to see this in the nervous system. Part of the reason why this is so difficult is that nervous tissue itself is actually opaque. You can't see through it. Light doesn't shine through it. If you look at a brain, for example, you can't see all the way through it, but we'd want to. We'd want to have a clarified brain. So people have, in the past, been trying to develop methods to take a brain that looks an awful lot like this and to clarify it, to make it transparent. If we can make it transparent, we can actually see structures that we would never have been able to see before. It'd be like trying to look at the CN Tower on a cloudy day versus on a really clear day. We'd be able to see the CN Tower really easily when there's nothing kind of occluding it or blocking our view, and this is sort of where we're at here. And we want to clear away those clouds. We want to make sure that it's a nice clear day to be able to see the structures that we need to be able to see. And a number of different types of techniques have been developed. You don't need to know all of these different names. Some of you, if you're doing your literature review, will see that, again, similar to what those 16th century astronomers were doing, were to develop different types of techniques. So some of these are CDB, Scale A2, 3-Disco, Cubic, and the one that sort of dominates is the one called Clarity. We're eventually going to come back and talk about this one individual, Carl Diceroth, but in the last 10 years, he's probably influenced neuroscience more than any other individual in the last 50 years because he's actually developed, and this is from his paper in Science, the capacity to make a brain go completely transparent. And he has a really interesting way in which to do this. But these are the reasons why we actually need to be able to do this. And you might think, wow, it's a really great trick, but why do we actually need to be able to clarify a brain? Can you think of what molecules actually prevent you from seeing all the way through? So here, the brain is actually there. It's as clear as glass in this particular case. What molecules in the nervous system actually prevent you from being able to see all the way through the brain? Yep. So the myelin is the biggest factor. So we need to get rid of the myelin somehow. We also need to get rid of a couple of other molecules, and we need to do that using all of these different techniques. And Clarity is one of those techniques that allows us to be able to do that. By doing this, we can actually look at entire tracts or entire groups of cells. These are just demonstrations. You don't have to know the data on any of these different slides, but it allows us to look at cell death as it's occurring in different types of tissue. We weren't able to see that before. It's only in the last 5 years that we can actually go back and view cell death occurring in the tissues that we've hypothesized and be relatively sure that it's not an artifact of the way that we're doing staining. So we can also go in and compare directly like the optical sections versus a cryo section. Here, I've actually cut a section. I've taken a loaf of bread and I've actually cut that loaf of bread, and this is what I'm showing you on the left. An optical section is different. I'm taking that entire loaf of bread, I'm making it go clear, and I'm showing you the same layer, layer by layer. I don't have to cut the tissue anymore, in other words, to be able to show you the exact same structures as I would if I had to go and slice every single piece of bread in that loaf. And this gives us this ability to be able to see structures much more quickly and in a much more logical fashion because we can see how they relate to other structures. I don't have to cut through all of these other layers and take away those layers. I can actually do this optically just by making the entire brain go clear. And especially when we're using fluorescence, this becomes a really interesting sort of method to be able to use. Now again, I don't need you to worry about all of the details here, but this allows us to do all sorts of different types of things. I want you to be aware of the technique. I'm not going to cover it in any detail, but we understand more about cell death and the different neurons that die based on lack of neurotrophic support, without having BDNF, without being able to activate RAS or other pathways, because of things like CDB or DISCO or 3-DISCO or Clarity 2. All of these different things, again, take time. It's not a trivial technique. In fact, when Carl Dijsselroth came out with his cell paper, it was actually reviewed for a very long period of time. And there was a lot of controversy because labs, for example, here in Toronto, weren't able to actually reproduce his results. So they took the tact of actually sending people, sending reagents to make sure that labs around the world, including labs here in Toronto, were able to reproduce their results, including things like Clarity. So again, it's a really amazing technique. Hopefully this gives you an idea of why we talk about cell death. It's not trivial. To be able to tell you about cell death in the next few slides, we need to actually develop these types of techniques to actually visualize groups of cells deep within tissue that we weren't able to do before. Do you have any questions about this? You don't really need to know the 3 or 4 slides that I just went through with you. It's for your own information. It's a technique that you may hear about in some of your 4th year courses, or you may hear about in graduate school, or some of you who are going into labs either in psychology or in neurobiology, you'll be actually doing these types of experiments because it's a really cool type of experiment to do. Any questions? No? Okay. So let me take you to this model, a little bit different. This is, again, sort of a model involving growth factors. We know, for example, that growth factors can come from neurons that surround this particular neuron here in the middle in panel A. So BDNF theoretically could be released here. BDNF could be released from this neuron and be retrogradely transported, etc. And this neuron, in response to having lots and lots of BDNF, will be nice and healthy. And one of the reasons why we know that this neuron will be healthy, if we were to take a section and cut it, whether it's optical cutting or cryosectioning, where we freeze the brain and then we cut these thin slices of 20 microns or less, we would actually be able to stain for things within the neuron, which is known as Nissl substance. Do you guys remember Nissl staining from before? So we can actually go in and see Nissl substance in a nice healthy neuron. We find it all throughout the cytoplasm. This is related to the ribosomes and the production of different proteins. This is in an intact system. And one of the things that was very popular, it still is popular to this day, is to do a surgical technique. If this is the neuron that might be actually producing the neurotrophic factor, it's releasing neurotrophic factor, and it's retrogradely transported, what would happen if we blocked the transport of neurotrophic factor? If we cut this axon, there's no way for the neurotrophin to actually come back to the cell body. There's no way for this neuron to receive the appropriate levels of BDNF. And I've just shown you in the last few slides that not having BDNF can actually lead to things like apoptosis. We know that this type of trauma is actually very, very common. So it's common in things like traumatic brain injury, where you can slice axons, or rotational injury of your brain. We know that you can do this physically by cutting groups of axons, as is shown here. Regardless of how we do it, when we do a model which is known as exotomy, there are some very characteristic changes that occur in this neuron. It starts to die, in other words. So one of the things that happens that we can actually physically see, and techniques like Clarity and CDB and others allow us to see this in great detail, the Nissl substance actually starts to disappear. The Nissl substance undergoes what is known as chromatolysis. It also causes the presynaptic elements to draw away. And all of these different factors lead to this neuron being isolated. It's like if there's bad news going on, everyone kind of goes away from the person that has all that bad news. And this is exactly what is happening to this particular neuron. The presynaptic elements retract away. Parts of the different glial cells actually physically will come in and will interrupt the synapse. The synapses are stripped, in other words. And in addition, we get these microglia that start to infiltrate, and the myelin sheath and the oligodendrocytes or Schwann cells also start to disappear. This neuron, by the way, also doesn't do very well. This neuron also starts to degenerate, and this was a particular model that was really, really useful. In the brain, this type of group of neurons is actually not that common. To find these three neurons in parallel, so neuron 1, neuron 2, and neuron 3, those are actually not very common to find. The other type that's much more common to find is where you have a group of neurons that actually innervate or make connections to a larger target tissue like muscle. So motor neurons, for example, do this. If you were to do this type of surgery, and this is the type of surgery that I did in my master's, it's actually fairly easy. So when you, for example, make an incision right under the ear, you're able to see visually, and you can actually see it in your own slides, the nerve looks like a white sort of thread that is in this opening. And you simply cut it. When you cut that particular neuron, all of these different things in that group of neurons actually occurs. The neuron undergoes chromatolysis. The neuron undergoes degeneration. The neuron has synaptic stripping. The neuron also will start to be infiltrated with macrophages and microglia, etc. Interestingly, because this is a motor neuron, do you notice anything different about this rat here in panel C? Anything that you recognize or see or can observe? Sorry? It's smirking. Not so sure it's smirking. So, anything else? Yes? Making? Missing an arm. Okay, we didn't do anything to the arm. It's a good guess, though. I like that guess. But no, nothing to the arm. Anything else? Yes? The nose is crooked. Okay. Maybe? It's a good guess. Yes. Alex? The eyes are always red in these rats. Yes. Okay. Good guesses. How about I just tell you? So one of the things, if you're looking very carefully, this actually recapitulates a condition that people have, which is known as Bell's palsy. If you damage the facial nerve on one side, that one side of the face will actually droop. A number of years ago, we had a prime minister named Jean Chrétien, and he actually had Bell's palsy, so one side of his face drooped. And you can see that the whiskers on one side are actually not going out as much as they are on the opposite side. So we know that we can cut these axons. We can actually see the effects of cutting these axons. And then we can see, can we keep these neurons alive? We don't have this support anymore. We've cut it. But if we were to apply BDNF, NT3, NGF, or other growth factors here, they would be a new source. And could they be transported back and keep these different motor neurons alive? And those were sort of the model that we used for facial exotomy. This turned out to not work out well, because when you were doing these types of experiments, these are the Nissl-stained brain sections. Each of these represents a neuron. And after exotomy, you can see most of them degenerate. So this is the normal side, not cut. And this is the side that has been cut. And you can see most of them are sort of degenerating away, etc. And all of these basically show the neurotrophic factors don't really do much. We had to drop those programs. BDNF, if we were to apply it in a damaged brain, actually don't do very much to the neurons that we want to keep alive. So in the 1990s and into the early 2000s, people moved away from this model. This was supposed to be the rescue molecule that everyone wanted to have. And again, I'm going to just skip over the acetylcholine and the choline acetyltransferase, but it's important to actually go in and look to see if the neurons are actually living through the Nissl staining that you see, as well as synthesizing different types of neurotransmitters. So let me take you through one other aspect that is really important as well, and this again supports a number of different ideas. And this is a really busy looking slide, so let me tell you what I'd like you to make sure that you know. One of the reasons why we don't think that adding extra BDNF or NT3. Even though BDNF is a really powerful molecule, and even though BDNF might work really well in terms of normal developmental growth, and it might be really important for maintaining synapses, and it might be really important after exercise, one of the reasons why those oligodendrocytes that we kind of just passed over and we didn't really spend a lot of time on, one of the reasons why people are more interested in those now in 2017 than we were before, those are much more complicated than we thought before. Remember, if we cut this axon, this oligodendrocyte starts to degenerate. It disappears, or the Schwann cell disappears. And we weren't really aware of why that was happening. We have a better idea of why we actually need these glia. It's not just for the myelination. Myelination is important. Myelination prevents us from seeing into the neuron or into the brain, but these oligodendrocytes actually play a really key role in being able to support these different neurons that you're seeing here as well. And the support comes in a number of different forms that I'd like you to make sure that you know. So this diagram is one that you should spend some time on. It will be related to what you get on your final exam. And one of the things that seems to be really important, the neuron and these oligodendrocytes are communicating with each other. So neurotransmitters are activating these oligodendrocytes that are wrapping themselves around this particular axon. These oligodendrocytes seem to be releasing things all the time, including trophic factors. So we lose those oligodendrocytes, we lose another source of trophic support. And we didn't really understand this for a very long period of time because these oligodendrocytes have a very particular type of vesicle. And this vesicle is called a multivesicular body, or an MVB. And these small, relatively small MVBs actually contain a lot of things. We used to think it was garbage, but we now know that this is much more important. These MVBs, or multivesicular bodies, can be regulated to release whatever is found inside of them, which include things like RNA, which include things like proteins, including neurotrophic factors, and they can be released much like you would at a synapse. So once you have neuronal activity, you would normally cause synaptic release. These oligodendrocytes release these multivesicular bodies. Sometimes this occurs in the form of things like exosomes, and these exosomes, again, also contain a particular type of RNA that we are now aware of called a microRNA. Have any of you heard the term microRNA before? Some of you have. These long non-coding RNAs that are wrapped inside of these multivesicular bodies, some of those get processed into shorter chunks. We don't really know all of the different details of these microRNAs, but I can guarantee you in the next 10 years, in your lifetime, and when you're doing research, these microRNAs are going to be implicated in almost every neurodegenerative condition, because we're now starting to realize that these microRNAs directly impact these neurons. They affect transcription, they will affect gene regulation, they will turn certain genes off, and they are much more important than we thought before. So when you're thinking about oligodendrocytes and just myelination, that theory, that's an old theory. That theory no longer is valid for you as neuroscience students in 2017. Myelin is incredibly important, but those oligodendrocytes are not just myelinating these axons. They are providing support to these axons as well. So I want you to keep all of that in mind. Know that exosomes contain things like microRNAs. And these exosomes are originally derived from these oligodendrocytic multivesicular bodies that contain a whole bunch of things, like long non-coding RNAs, as well as these different proteins. Again, a really important field that many of you might be encountering as you do your literature reviews. Do you have any questions on this before I go on? Anything I can clear up for you, or things that you might have read about while you're doing your lit review? Yes. Yeah.
So I don't understand. Are the MVBs, are they made up of microRNAs or are they
So, So these multivesicular bodies, they're relatively big. So they're much, much larger than you would have for these small vesicles. So when you see those pictures of the presynaptic terminal and you get these small vesicles, these are much, much bigger. So they're about 100 to 1,000 times bigger. These then get processed and they will release smaller versions of themselves. And these smaller versions are known as exosomes. So these are exocytic bodies or exosomes. And we know that somewhere in between this process that might involve things like calcium, that the long non-coding RNAs and proteins that are found here, they get processed into these smaller microRNAs because microRNAs are derived from the bigger long non-coding RNAs. So we think that the exosomes are a direct derivative of these multivesicular bodies. We don't understand all of that process yet. In fact, it's only about the last 6 or 7 years that people have started to pay attention to microRNAs within the brain. Any other questions or things I can clarify for you? No? Okay, so again, just to reiterate a couple of things, I don't think that we need to belabor this point. I'm primarily interested in looking at apoptosis, and some of you who have taken other courses will know that neurons usually die either through programmed cell death or apoptosis versus necrosis. You know that in stroke, for example, necrosis might be really important in some areas of the brain, and apoptosis is important in other areas of the brain. The reason why we are interested in apoptosis, or this programmed cell death, is because it requires the expression of certain genes as well as proteins. We have to turn them on, in other words. That means that if we interfere with this process of apoptosis, we could prevent cell death, and that's why we're interested in it. Necrosis is, again, very, very different. It doesn't seem to require the expression of new genes in order for this to occur, and it usually is occurring in a wave. Large numbers of cells are dying rather than individual groups of cells at any given point in time. You don't have to memorize this slide. Most of this is going to be repeated for you in the next few slides. We can characterize this, and you don't have to memorize this, in a number of different ways where these small apoptotic bodies originate from the neurons as they start to die. And again, it's just to demonstrate that morphologically now, based on different types of stains that we can do, including using clarity, we can actually watch this process occurring almost in real time in a clarified tissue. So we can actually see that this process does indeed occur during early development. After we damage neurons, etc., we can actually observe apoptotic bodies forming versus the form of necrotic cell death after things like stroke. So again, don't try to memorize this slide. Don't worry about this slide here either. But this slide here, I do want to spend a little bit of time on, and I do want you to understand a couple of things related to this. So we know that apoptosis actually involves a program of cell death. We need to activate genes. We need to activate different proteins that derive from these different genes. We know that this is also really well conserved. In fact, the first place that we see programmed cell death is in C. elegans. And we know that we, as mammals, also have many of the same homologs that we see in C. elegans. If we have the same if C. elegans has the same processes that we do, this is a really important mechanism that runs across a number of different species. The ones that I'd like you to be more familiar with, and I know many of you have had these in other courses, and in some of you might have not had them for a while. The ones that we're the most interested in within the nervous system are the ones that in C. elegans are known as SED3. In mammals, it's known as a caspase, in particular caspase 3, etc. And these caspases are cysteine aspartate proteases. They cleave at these specific amino acid residues. And these are involved in cell death and in apoptosis. And then we know that there is another group, which is known as SED9, derived from the C. elegans family SED9. In the mammalian homologs would be known as the BCL2 family. And it turns out that in the BCL2 family, that if we're to take a look, some of these will cause death of neurons, and some of these will actually protect neurons from dying. And again, our hope was that different neurotrophic factors would influence the increased production of BCL2, and eliminate or reduce the levels of things like BAX or BIM or other proteins like BAD. So all of these pro-death ones are ones that we don't want to activate, and we want to keep BCL2 or BCL-XL levels really high. The SED4 you don't need to worry about, because in the next diagram I'm hoping to kind of link all of this together with you. In the mammalian nervous system, the most important members of this apoptotic cell death pathway are the ones that are sort of outlined here as caspase 9. So this caspase 9 is the one that we think is important because it will be a key factor in activating caspase 3, and it's really the caspase 3 that will cause the neurons to die. In neurons, we're not 100% sure, but we know that many forms of apoptotic cell death involve the mitochondria. And the reason why this whole cascade begins is somehow the mitochondria loses its ability to hold on to one molecule, which is known as cytochrome C. So a neuron might not be able to hold on, the mitochondria in a neuron might not be able to hold on to cytochrome C if BDNF suddenly disappeared. Then suddenly now, caspase 3, I'm really messing up, so apologies on this. Cytochrome C might rush out of the mitochondria. Cytochrome C will then activate this caspase 9 pathway, and this might in turn activate the caspase 3, leading to cell death. Remember, caspase 9, for those of you who are thinking, some of them are apoptotic and some are anti-apoptotic. Some cause cell death, some don't. Obviously, in this case, we're looking at the factors that cause cell death in that caspase 9 pathway, and this is what we think happens in neurons. Neurons need a lot of energy. We know that they have a lot of mitochondria, and as a result, we know that they have a lot of this cytochrome C. If the neuron at any given point in time releases too much cytochrome C, this default pathway that's ready to go at any time becomes activated. We're not sure of the link between BDNF and other neurotrophic factors that normally prevent the mitochondria from releasing cytochrome C, but this pathway is pretty well described. We're reasonably sure that this is what causes cells in your brain to die after things like stroke or after you cut a group of axons. This pathway is the one that becomes activated. Do you have any questions on this before we go on, or any questions in general? No? Okay, so again, we're looking at a number of these neurodegenerative conditions. The models that we used to look at, where we thought we could rescue neurons, protect neurons, keep neurons alive by adding these growth factors that we've identified, doesn't work. That has come and gone. Nowadays, we don't look at neurotrophic factors in the same way. We want to maintain the level of neurotrophic factors through things like exercise, etc. But right now, we don't really have a good way to address this neurodegeneration. We're kind of leaving that up to your generation to be able to address this problem and to reverse this neurodegeneration, but we don't have a good method right now to be able to do this. And we know that there are a whole host of different diseases like Parkinson's, Alzheimer's, ALS, you name any neurodegenerative condition, ultimately it involves their apoptotic loss, and we don't have a good way to be able to regulate this. So unfortunately, that's sort of a downer note in terms of what we can do and what we can't do. We have a number of techniques that might be able to address the basic mechanisms. Things like Clarity will help your generation understand how these neurotrophic factors work and to be able to identify cells as they're dying and potentially rescue those cells. But we don't have a really good way to be able to stop or reverse this neurodegeneration right now, and most of you are aware of that fact, that that's a limitation. We don't have a way to cure Parkinson's or to stop Parkinson's from actually progressing once we've identified it as Parkinson's. So any questions before we switch gears just a little bit? No? If I were to summarize most of what we've been talking about up until this point in time, I think you would all agree. We've been looking at a lot of controversies related to neuroscience. So controversies related to parabiosis and whether or not age-related factors, rejuvenation factors work, stem cells, exercise, all of those are relatively controversial. Do you think there is an area in neuroscience where there's no controversy? And can you name me one area in neuroscience where there's no controversy? So every area has like tons of controversy? No? Tell me which one doesn't. Any ideas? Sorry?
That's like a really vague question. Maybe there are some things that are settled, but like
So
other
you've taken other neuroscience courses and psychology courses and cog sci courses. So what's the one thing that you keep learning about all the time in every one of your courses?
potentials.
Okay, no. It's not action potential. There's no controversy there. Okay. Any other areas that you don't feel that there are some controversies? Are you all confident that learning and memory has no controversies associated with it? How many of you can tell me like the pathways that you think are involved in LTP and synaptic strengthening and plasticity? All good? All good, right? The fact is, memory is one of those things that we're going to talk about because we know BDNF, for example, enhances memory. We don't understand all of memory. So we don't really know, for example, if I were to ask you right now, this instant, think back to your grade 9 teacher. Can you tell me the name of your grade 9 teacher? Probably, right? And if you're thinking about that, one, I can't tell you how you retrieve that memory. I don't know what groups of neurons contain that memory. I don't know how you retrieve that memory. I have no way to tell you how that process occurred. I think there are some structures that I might be able to relate to you regarding that particular phenomenon, but we make a big deal out of learning and memory. But in fact, we don't know very much about learning and memory. What I'm going to show you, not in this lecture, but in the next lecture, there are some really big areas that are developing. So let me ask you a really fundamental question because it's going to be important in the next class. If you were to look at synaptic plasticity, the strengthening or weakening of synapses, do you think it's better in you or better in someone who's 3 years old? And why do you think that? So who's got more synaptic plasticity? Someone who's 3 years old or someone who's 20 years old? 3 years old? Why do you say that a 3-year-old has more innate synaptic plasticity than someone who has been using those synapses for like 20 years? Why? Your answer is correct, by the way, but why? So we're going to talk about this. This is a very controversial field related to infantile amnesia and related a little bit to the stabilization of synapses and proteins that stabilize synapses. But I wanted to actually go into a little bit of the details related to memory. And memory is one of those things that is really, really bizarre. Memory is not something that we really understand well. We're trying to understand it. In fact, there's a lot of research going on into memory, but we don't really know all of it. In fact, we're making a lot of guesses about learning and memory. So when you're retrieving a memory, where that sits in your brain, no idea. Multiple synapses in your brain? No idea. How that got there? No idea. So I'm just going to tell you about the best guesses that we're going to look at. So many of you will have heard of Eric Kandel, and I'm going to go through why Eric Kandel is so important in today's lecture. He's actually really important for us as neuroscientists for a variety of reasons, not only for what you think he's important for, and I'm going to outline that for you today as well. And I'm going to go through all of these other things. So before we get there, let me ask you a couple of things related to this, and then we'll take a quick break. So if you can log on to the polleverywhere.com site using the number that you see up here. You'll just have to wait for my question, so if you're all on there. Who's doing that? Okay. Give you the question. Okay, so who is Classic Kirk on this picture up here? Thank you. Okay, so at least you're paying attention. How many of you would guess that in A, this is a scientist by the name of James McConnell? Yes? If you think he's James McConnell, put your things over there on A. If you don't think he is, put it where you think James McConnell actually is. Okay, so most of you are saying that A is James McConnell. How many of you now, so great, thank you for doing that. How many of you actually know who James McConnell is? No. Okay, so you've correctly identified him. That is indeed James McConnell. Fantastic. Stop with the whole thing on there. Okay, let's just go out of there. So James McConnell is actually really important because he developed a model system. And this model system is here on the left. So this model system that we're looking at, you have a choice. You tell me what the model system is. And how do I, there we go. So you tell me, is this model system that James McConnell developed? What are we looking at here? Because this is James McConnell's model system. Okay, so just give it a couple of seconds. Yep, a couple more seconds and then we'll kind of wrap things up. So James McConnell actually was the first individual who actually caused problems in neuroscience. We entered the golden age of neuroscience in the 1950s. 1950s, we developed drugs, we understood the brain, we looked at different structures neuroanatomically, and then the 1960s came along. And in the 1960s, James McConnell actually reversed neuroscience in a really bad fundamental way for a variety of different reasons. And one of the reasons was that he used this particular model organism that you are seeing here. And this model organism, by the way, is known and I'm actually going to get to it in a few moments, is actually a planarian. How many of you have heard of a planarian before? So these are often found in fresh water. If you were to go to High Park and you were to go down to the pond and you were looking near the edge of the water, you would actually be able to see these planarians near some of the different plants there. And he used these planarians for a really specific reason that we're going to get to, but we'll take a break first. Okay, so and we are going to try to do a movement break, so if you can just hold on for a quick second. Since this is our last movement break before we actually go on break, if you can, this is a little bit more energetic. So we're going to try to stand up and we're going to try to go through this. Come on. Okay, so a wonderful movement break. We're going to get you off your seat and moving.
It's dancing. He's coming down too. Personal space. Yeah, a little bit more. Oh, the feet too. Oh yeah. Okay. It's like salsa. Yeah. Well, not really with the arms. Over there. This way. Oh, the hips. Don't forget the hips. I would never forget the hips. Come on. Yeah, this is like salsa. This is salsa. It's merengue, apparently. No, it's mixed. I guess so. No? Okay, this side, I'm not sure. What's bounce? This is an alternate to the Macarena. If this was a dance competition, I would be losing. It's the other way. This is nice. The trick is to have a light foot. Yeah, it's something alright. Yeah. This is why I'm not good at DDR. Oh shoot. Oh my gosh. She's doing this so fast. I wanted to do that. Oh wait, she's doing it wrong. Well, she's not turning at all. Alright, well, the other way. Are you sure? You predict your movements. She's gonna clap now, right? Oh wait, no, never mind. You can clap, yeah. I'll come down this time. Oh, something. Okay, this is new. Did we? Oh, yeah. I don't remember. What? Oh.
Alright, thank you everyone. We'll take a 10-minute break and come back. Thank you.
It's just very helpful.
So it stays. How is it that I see that but not? Oh, which is fine. Like I see it on. Wow. Okay.
I see what you mean now. It stops working.
That's okay. I don't really care as long as it's maybe clear.
Is clarity? So for you still have to modify this. Because like
It's just a diagram. In fact, it kind of loops around.
So it's not just that one side.
It is. But the cell body, remember, wraps around the axon.
What about Bell's palsy? How does, because I know it's damaging the vision. Right, so,
It's viral.
oh, so how does it recover? How does,
It doesn't. It dies.
oh, it does. Because I knew someone who had Bell's palsy, but he regained like the muscle. So
But the nerve is never going to come back.
so it uses like other means of.
Watch the plant start like shaking. Yeah, but I'm curious. Plants have the same watch it grow. Throw my own. Asked me that. So I asked you that already. I asked everyone that too. Again. 2 hours.
Is something not working?
It's working, just need to make sure. Okay, I didn't turn it off. Good. We're good. I thought I turned off the sound.
Okay, if you don't mind, we're going to get going. So just before break, I mentioned to you that in the 1950s, and we've looked at neuroscience that developed in the 1950s. This is where Wilder Penfeld is exploring through surgical means and mapping out the brain, figuring out where the sensory motor cortices are, stimulating. All of that's in the 1950s. We know the classic drugs that we still use to this day are developed in the 1950s, and neuroscience is entering into this golden era where neuroscience is advancing. At the very tail end of the 1950s, we have a number of really interesting experiments that are going to set back neuroscience for the next 20 years. And this actually becomes a really interesting proposition. So we have a neurochemist in Sweden in 1959, Holger Heiden, and he's kind of interested in what we just talked about, learning and memory. Learning and memory has fundamentally driven all of the great expanses and great advances that we've seen in neuroscience since we've been examining the brain. Memory is important, how you learn is important, and I'll get back to that in a few moments. So Holger Heiden proposes in 1959 that there has to be something in a neuron that actually captures memory, the essence of memory. So I've come into this room, I've just sat through this lecture. There has to be a way for me to remember something that I've learned in this lecture, and he proposes that it has to be stored in a molecule. Now keep this in mind as we go through the rest of today's lecture. And he decides that out of all of the different molecules that he knows in 1959, DNA has been published, and DNA now, the structure of DNA is well known, and he knows that their proteins exist, and he also knows that they're RNA. Out of all the molecules that he sees, the one that makes the most sense in terms of being able to quickly and rapidly modify itself as you are learning something new is RNA. That's the only molecule that can actually change its overall structure based on what he knows in 1959. So the rapid modifiability, you learn something new, something in your brain or in your neuron has to change quickly. The only thing that can do that for Holger Heiden is RNA. So he's proposing that RNA is this memory molecule that makes a neuron learn something brand new, and it's what he thinks is important in memory. He decides, like any good scientist would do, to test out his hypothesis. So he trains a group of rats to be able to climb up a wire, and as they climb up a wire, they're learning a new task. They don't normally do this. They have to climb a certain distance to be able to get their reward. They have to learn something new, in other words. So he wanted to know if these rats, before, or the control, before learning and after learning, are there any changes to the structure of RNA as this rat is learning? So again, he's working with a colleague in 1962. Holger Heiden and Egehazy decide to do all of these different changes. Look at the place where he's examining. So we would look at the hippocampus, for example, in terms of learning and memory. But he decides, because this is involved in spatial balance, as the animal is learning to climb this wire, that he's going to look in the area of the brain that's involved in balance. So you have to climb a wire, you have to learn something, and his idea was that he would look in the vestibular nucleus, which is involved in a form of balance regulation, and he wanted to know, are there any changes? So after the animal has learned to climb this wire a certain distance, he goes in, takes out their vestibular nucleus, and he realizes that, wow, you know what? One, after they've learned something, the vestibular nucleus, the amount of RNA in an equal amount of the vestibular nucleus before and after actually goes up. In addition to that, and more importantly, he decides that, based in 1962, that the RNA has indeed changed its fundamental base pair sequence. Now, think about that from your perspective in 2017. You know that that wouldn't be possible. The RNA can't really modify its sequence from what it is before to like having a brand new sequence. But that's his hypothesis, that somehow, as this animal is learning, it's storing that information in RNA, and that RNA itself is intrinsically, by itself, changing the base pair sequence to encode for that memory as it's learning. We would laugh at that theory now, but this was widely accepted in the 1960s. This became really important for one reason. He had identified a molecule that was actually strongly associated with memory. So it may not be right, but this was sort of the trend that he started. So memory, in theory, could be stored in a brand new way as one single molecule within a neuron. This again was a really attractive hypothesis, and this idea was taken up by James McConnell, the person that we were just looking at. And James McConnell decides that he wanted to look at exactly the same thing. He wanted to actually look for an RNA memory molecule. He wanted to use a planarian. So a planarian is that flatworm. This is what a planarian normally looks like. And he wanted to look at planarians, not rats, but he wanted to look in planarians for a model of learning as well as memory. Planarians are intrinsically afraid of light, for example, so they will actually learn to move away from light. And you can actually train them, giving them a food reward to move against their normal sort of way of dealing with light, to move toward a light if you give them enough of a food reward. So you can train them to move in the directions that you want, and you can train them to move either to the left or to the right, depending on whether there's a reward, whether there's light, stimulation, etc. So is the animal learning, and does the animal remember? Yes. Meeting all of those criteria. But James McConnell doesn't do the same experiments that Holger Heiden and Egehazy does. He actually does something completely bizarre, and this throws back neuroscience and science in general for a number of different years. Years. He decides that, hey, if there's a molecule in the nervous system of this planarian after I trained it, what I'm going to do is somehow isolate that molecule. It might be RNA, it might be a protein, whatever it is, that memory has to be stored as a molecule. If I take the brains of these planarians, or if I take the nervous system of these planarians, and I ground up the nervous system of the planarians that have just learned this task, and I feed the nervous system of these planarians to ones that are naive, they've never been trained before, you're basically causing them to be cannibals. Will they actually take that molecule that's in the ground up nervous system, absorb that molecule, and absorb the memories along with it? Does that sound like a great idea? That sounds like a great idea. Wouldn't that be fantastic? You actually don't have to learn anything. All you have to do is find someone in fourth year or in graduate school, convince them to give up their brain, grind it up, eat it, and you would have all of their memories. That sounds very attractive to me. And this is sort of the idea that he had, that he was able to demonstrate in his papers that by doing so, by taking the nervous system of these trained animals, grinding up these nervous systems, and then feeding it to these naive planarians, the planarians that had never encountered this training protocol before, if they're given a reward, they will move toward the reward, or they will move away from a light, just like they were trained, just like the animals that of the brains that the trained planarians had before. So he was able to demonstrate that all I have to do, I don't need to train these animals, I just need to give them the brains of animals that have been trained. The memory molecule is passed along. Do you like that idea and hypothesis? Does it sound rational? Does it sound logical? Doesn't, but he's able to show this over and over again. Can you come up with a hypothesis to explain how when I take that animal that's untrained and I put them on the same dish, that they're able to move in exactly the same direction that they should? Any ideas? Any hypotheses? The problem is he used exactly the same dish that he used before. So they are put in a dish. They're learning to move either to the left or to the right. And now you take this planarian that's been fed the ground-up brain of the previously trained planarian, and now they're in the same dish. So he wasn't able to account for the fact that they leave chemical trails as they're moving along on these dishes. So one scientist realized that this is the reason why they're seeing these untrained planarians moving in the same direction that they should in these training dishes. This whole field goes into disarray. No one wants to do this because he falls out of favor, and as a result, people decide, we're not going to look at molecules related to learning and memory. That's not going to work. We'll never find it. This area of research goes into hibernation for 20 years until another individual comes along and decides that maybe there's something that we should actually be doing related to these different molecules related to learning and memory. And this individual is Eric Kandel. And one of the reasons why I said Eric Kandel actually was more of an impactful figure than we give him credit for is because he actually reversed that trend. People were staying away from trying to identify molecules related to learning and memory. And Eric Kandel comes along, and he wins the Nobel Prize in the year 2000. And I remember I was at the Society for Neuroscience where he was a keynote speaker in the year 2000. And I remember most people were talking about what did he actually do to deserve the Nobel Prize for his work in aplysia. But he actually reversed the trend. He actually came up with a plan that we still use to this day. So after 17 years, we're still using the same fundamental questions. These are the questions that he asked. You don't have to worry about memorizing these. I'll tell you what you need to know in the next few slides. But he wanted to know, is there a cellular mechanism? Not the type of mechanism that James McConnell or Eggy Hazy or any of those others were doing. Where is this circuit in the brain where all of this is occurring? How is memory stored? Like, what is the actual physical mechanism by which we store memory at these different synapses? And he wanted to know this. He is still not able to answer this question, so it still remains as one of the unsolved questions. We don't really know how memory is retrieved. So when you're thinking back to that person that you haven't thought about in over 10 years, we don't know like what pathways you've reactivated to be able to cause that memories to be retrieved. We know that there are processes now that interfere with learning and memory. Do you know any processes that interfere with learning and memory? Anything? Sorry?
Lack of sleep.
Lack of sleep. Definitely lack of sleep. Yes. Alcohol. Definitely alcohol. Anything else? So sleep. Don't drink alcohol before you show up to your term test. Anything else that will help? Yes? Attention. Paying attention to be able to strengthen synapses in the first place. And I think it's one of those fundamental features that we are all really interested in. I can tell you one other thing that we're going to talk about a little bit later on. Every time you reactivate a memory, that memory trace, the engrams that we're going to talk about in the next lecture, become reactivated. We don't know how they become reactivated. We don't know how they're retrieved. But during that period of time, they're what we call labile. They're able to be changed. Now, I'll give you a really, really good example of this. So my wife and I were talking a few years back about how we met and how we were getting married. And so we were talking with some friends, and I remember this in great detail. I remember every single minute of how I proposed to my wife. So I remember I was on the beach. My wife was like walking with me. I was holding her hand, and as the sun was setting, I remember looking at her and saying, "Do you want to get married?" And I remember that in great detail, right? And then my wife looked back at me and said, "That's not how it happened." So we were actually sitting in a restaurant. We were having steak, and we were talking, and then you decided that you wanted to get married. One of us is lying, or one of us misremembers. But to me, that memory is actually really, really vivid. The problem is that's actually a scene from a movie. And I actually don't have that memory. I think my wife is actually right. So every time I reactivated that memory, I was actually allowing other memories to sit on top of it and to reshape some of those memories. And this is why we know that memory is one of those fundamental features that we need to be able to understand from a cellular perspective. So one of the great things that Eric Kandel did, and many of you will know his experiments, I'm not going to go through his experiments, is to simplify things. So he decided the human brain is just too complex. I can't, even though I'm a clinician, and he is a clinician, even though he's a clinician, he decides the brain just too complex. I need a simpler organism. I need a simpler circuit. And this is really what we try to do in neurobiology all the time. We need to understand it from a simpler form. We need to understand it at a basic level instead of trying to understand the entire brain at the same time. So this often means that we are using invertebrates. Eric Kandel uses aplysia. People use C. elegans. Other people use slices of the hippocampus. And sometimes that means that we're also using cultures. And one of these cultures that I do want you to make sure that you know are these really specialized types of cultures which are known as autopsies. And I'm going to get to that in a few moments. So this is a normal dissociated culture. So I will take an embryonic brain. I will take it out of the mother. I will take the embryo out of the mother. I will dissect the brain. I will dissect an area of the brain that I'm interested in. Usually we pick the gestational age of embryonic day 18. That's the easiest one for us to use because most of the structures in the brain by embryonic day 18 in a rat are fairly easy to identify. I can identify the hippocampus really easily. I can identify other structures in the brain easily at E18. So we take these embryonic brains or regions of these brains and we dissect them. We simply pull them apart. We get them down to the single cell and then we put them on glass or we put them on plastic. And by having individual cells, and these are individual cells, and if we have the right growth factors that are added to them, after about 7 days in vitro or 7 days in culture, so we take them out on day 1 or day 0, we put them on these specially coated plastic or glass slides. After 7 days, this is what the neurons will look like. If we continue to allow them to grow, they put out these really long processes. And you can see some of these processes here, and they will look a lot more like neurons. And we can actually keep them alive for a fairly long period of time. But can you identify single synapses here? It's really, really challenging. So even though this is a simplified system, and this type of dissociated culture is a simplified system, it actually is fairly convoluted. So this one neuron, for example, may be receiving like hundreds of different inputs from 100 different nearby cells. And that becomes challenging for us. So some scientists have actually decided that I don't need all of those neurons. I just need one neuron. I need one neuron and synapses. And so what they decided is to create what is known as an autops. And I do want you to know this model, and I do want you to know some of the advantages and how to confirm that you've actually developed an autops. So remember, we're always trying to simplify things, whether it's using an invertebrate model system, whether it's using fewer neurons, and the simplest type of synapse is known as an autopse. So this is a very, very special type of cell culture. So we do exactly the same thing. We take out embryonic brains or regions of brains, and instead of just putting them on specially coated plastic or specially coated glass, we actually have to punch the glass, or we have to make an imprint on the glass. And this is the type of device that we use. We will physically go in and stamp these little cover slips, and by doing so, these little ridges allow for these little bumps, and on top of these little bumps that we print onto the cover slip, we will have one single neuron growing on that one single little bump, and you can see these individual little bristles. They look a lot like a comb, for example, or a brush, and on the ends of those, you would actually be able to grow a single neuron. And the single neuron will actually make, because there are no other nearby neurons, they're all growing on their own independent islands, it will actually make a synapse onto itself. So it makes its own synapse onto itself. Instead of sending out a synapse to a nearby neighbor, it will actually connect to itself. And these are perfectly fine synapses. These are the simplest types of synapses that we're able to find. So for example, we could see the presynaptic markers that will stain for the neurotransmitter vesicles for glutamate. So vGlut1 is normally a presynaptic marker. The proline-rich synapse-associated protein, or ProSAP1, is the postsynaptic marker. And we can see that the presynaptic and postsynaptic elements actually lie on top of each other, just like they would between two other neurons. Now it's just on this one neuron. So we get presynaptic and postsynaptic all occurring within this one single neuron. And we can go in and see that these are actually making these connections on these dendrites. So you should make sure that you know how an autoptic culture is formed. So we use these punches. You should know the advantages of using an autopse. So it's a very, very simplified form of a synapse, and that we can go in and look for both presynaptic, postsynaptic, as well as dendritic markers to show that this looks an awful lot like a normal type of synapse that we would see elsewhere within the brain. So this is a really simple system, and this autopse is the simplest system that we could possibly come up with. But there are some other simpler systems as well. Can you think of a reason why we might actually want to use an intact organism? So this is a simple synapse. We can study it all day long. We can look at action potentials. We can look at synaptic release. We can even look at LTP and strengthening. But why might you want to use an intact organism? A simple intact organism. Any ideas? Stephanie? Right, so a system-wide approach, being able to see behavior as a result of modifying a circuit. So these are some of the advantages, and again, you don't have to worry about memorizing this list, but these are the types of things that we look for when we're looking at a simpler system. So we need fewer neurons. That's a given. We can't identify like 86 billion neurons in your brain. Makes it really hard to find things happening. So a simple small nervous system, we need large neurons. Large neurons make it easier for us to identify, to see, as well as record from. We also need to know what those neurons are. Are they excitatory? Are they inhibitory? We need to know that information. We also need to know, are they part of a circuit? So nowadays in neuroscience, we look at what is known as the modern circuit theory. We have about 8 different nodes in your brain, and they form different circuits between the different nodes. And that's relatively new as a hypothesis of what causes different types of behavior. And we also would like to have simple genetics. So C. elegans has a very, very simple type of gene organization, and we know all of the different genes that are found within C. elegans. Those are the types of things that we want. However, Eric Kandel and his collaborators actually didn't go with any of those. Many of you will know that he went with a Plisia, this large sea slug. And this large sea slug also has some innate advantages that are available were available for him as well. And one of the biggest reasons why this is really important is if you were to examine the sea slug, you have an identifiable circuit. So you can identify each of these different neurons, which make up a really, really simple circuit. You don't have to memorize this circuit. I don't need you to memorize the different types of interneurons, the motor neurons, or which muscles that it's innervating. None of that's important. It's just to give you an idea that you can look and identify circuits within aplysia. Based on all of this, he was able to show very, very uniquely that he could see changes in modification. So he could see strengthening of synapses. If you kept habituating a synapse, he would be able to see changes at the level of the synapse and the behavior. And so this meant that he was changing synaptic transmission. He was also able to show that this was the result of different molecules like protein kinase A, which was then working on different channels and different receptors. He was able to show a cellular basis. And he also was able to show that memory was stored within the context of these synapses as well. So Kandel was able to reverse that trend. And that's the principal reason why he was awarded that Nobel Prize, that he was able to answer all those questions that people like McConnell and Egihazy and Heiger-Holden actually had set neuroscience back. So he put us back on the right track to be able to identify these different systems related to how synapses can be modified and how modifying synapses can lead to learning as well as memory. So we're going to be talking about this in some more detail over the course of the next lecture. And I'm going to be telling you a little bit more about LTP. Now, again, you don't need to memorize these slides. This is all of the different blueprints that we're going to be using in the same way. I'm going to be telling you about how we strengthen synapses. And many of you will have heard of LTP, and I'm going to very, very briefly talk about LTP in the next lecture because it becomes important in our discussions about developmental forms of LTP and strengthening of LTP. For those of you who have had lectures on LTP, have you ever thought about this? Because this still drives the synaptic biologists a little bit nuts. You've heard that synapses can get stronger, right? Yes? Why don't they just keep getting stronger? Why do they have to stop? So no explanation. We don't know. We don't know if there's a set number of space that proteins can actually occupy. We don't know if we can keep growing synapses beyond a certain point or if they have to split after a certain point. We don't understand all of those rules. But they always have a discrete level. We can't really continue to modify these. And it was only until 2017 that we actually understood why this rule actually applied. And there was a group at Stanford University that actually was able to solve why this is. And if you're really interested in it, you can come and talk to me because I'd love to talk to you a little bit about what Daniel Madison and others discovered back in 2017 or early in 2017. So a couple of other things that I wanted to mention to you. Many of you will know about the hippocampus, and this is where we're interested in learning and memory. There is an input to the hippocampus, and we know that the hippocampus is only involved in short-term memory. The hippocampus is not involved in long-term memory. For you to retrieve those memories from 10 years ago, the hippocampus was really not that important. Memory isn't stored in the hippocampus. We're well aware of that. We've known that for well over 20 years now. But we don't know all of the essential rules of what the hippocampus is responsible for. We know some of this from HM, or Henry Molaison, and many of you will have heard his story. Again, really fascinating story. Can anyone tell me his story like really quickly, over like 30 seconds? What happened? What happened to him? HM, second most famous patient in medical history. Why is he so important? What happened to him? Andy, you're dying to tell me. So he had anterograde amnesia. Why?
Because his hippocampus was damaged, so he can't recall anything what happened.
How was his hippocampus damaged?
I think he had epilepsy.
So he did have epilepsy, but that didn't cause him to actually have anterograde amnesia. Sorry? Right. So William Scoville actually was a neurosurgeon down in Boston who did this surgery. This is actually his brain. Interestingly, this type of surgical procedure, if you have different forms of epilepsy, this procedure is still done. Do you think that's a good idea? There's one difference though. So what do you see missing from his brain? It's not a trick question. His hippocampus is missing. How many of his hippocampi are missing here in this picture? Both. We still do this procedure, or surgeons still do this procedure, but they don't remove two. They only remove one. So we need one hippocampus. One hippocampus is enough to prevent anterograde amnesia. But this procedure is still done for different forms of epilepsy. You'll hear about that in some of your fourth year courses. And it becomes really important because now we understand the essential role of the hippocampus. So we know that the fundamental cellular property of why learning and memory begins in the hippocampus, especially from Henry Molaison. We know that he had this anterograde amnesia. The hippocampus isn't where we store memory because he was able to remember things from many, many years before he had that surgery. He could recall things from before the surgery, but not after the surgery. That's because these memories were consolidated elsewhere. And in the next lecture, we're going to talk about consolidation, what happens in when you're young versus what happens when you're older, and how this consolidation of memory is thought to occur in these memory engrams. And we know that the hippocampus is really involved in very short-term forms of memory, which we call LTP-enhanced memory. So LTP gets you a certain way. We strengthen certain groups of synapses within the hippocampus, but that's not enough. That only lasts for a certain amount of time. And for a long-term memory, where you're remembering someone's name for like 10 years, 20 years, etc., we actually have to remove that memory from the hippocampus and put it elsewhere within the cortex. And this hippocampus is only involved in these relatively short forms of memory. So we want to know this long-term memory formation. We know that there is a circuit that's involved in this process. To be able to generate these memories, we have to move these synaptic strengthening molecules somehow up into the cortex. And this is what we'll be talking about in the next lecture. And we know that this information probably leaves from the hippocampus through the fimbria fornix. So there are a lot of experimental evidence that is coming out now that if we can stimulate the fimbria fornix, we can actually cause long-term memories that have been buried in your brain to be reactivated. So this pathway that exits the hippocampus is thought to be really important, this fimbria fornix. And these short-term memory types of synapses are thought to get up into the cortex via this structure, which is known as the fimbria fornix, that I'm going to show you in the next few slides. So you can actually damage memory, not just by damaging your hippocampus, but by also lesioning the pathway, which is known as the fimbria fornix, where we have axons leaving the hippocampus, which is collectively known as this fimbria fornix. So again, we're going to be talking a little bit about this strengthening or weakening in the hippocampus, and in particular, we'll be looking at plasticity related to LTP and how this is involved in the long-term memory changes that we're interested in. And I'm going to just skip ahead to one particular slide because I'll come back to LTP a little bit later on. I need you to understand this overall flow. So this is a structure that you'll see several different times, the hippocampus. You need to, as your colleague mentioned, you actually have to be paying attention to something in order for you to remember it later on. And we know that we get all sorts of sensory signals. You're looking at something, you're listening to something. You have to be paying attention for you to actually begin this process. These integrated sensory signals are entering into this hippocampal formation through the first part, which is known as the entorhinal cortex. The entorhinal cortex and cells within the entorhinal cortex will send their axons up into the next part of the hippocampus, which is known as the dentate gyrus, and then it will send it into the CA1 and CA3 and CA1 regions of the hippocampus. And the axons here will leave and enter into the cortex through those structures known as the fimbria fornix. So I need you to make sure that you know the overall neuronal architecture and the anatomy associated with this. The parts at the top you don't need to worry too much about, but I do need you to know where integrated sensory signals first come into the hippocampus and how in the hippocampus, so all of these structures here, how they are related to each other. Not only that, I need you to go back and review some of the structures that are related to the hippocampus, and this becomes important. You don't need to know the next few slides. We actually need to know this structure for a reason. When we are going to be talking about how we stimulate this in a dish or how we actually stick in electrodes, we can actually see this under a microscope. So we do this. Some of you might be going into fourth-year labs or third-year labs as part of your research projects. You'll be asked to do something very similar, to cut a slice of the hippocampus, and then from this slice of the hippocampus to be able to stick in an electrode in a particular area of the hippocampus to stimulate it to cause LTP, which underlies learning, which underlies memory formation. So this is what it would look like. You're able to see all of these different structures under a microscope. It's not a very powerful microscope. It's just a dissecting microscope. Do you know what this structure down here, this nylon net, what is that? It's a really high-tech piece of equipment. You'll often see it if you're walking through an electrophysiology lab. Any ideas? Where do you normally see nylon nets? Probably the most embarrassing thing if you're a neurophysiologist to actually work on. Any ideas? Do any of you do electrophysiology? No? Okay. So this was, the first couple of times it was really embarrassing for me. You have to actually go in and buy pantyhose, stockings, and this is the nylon net. We create this. The slice sits on top of this nylon net, which is really pantyhose, and someone usually gets sent out to buy these things. Often it was like me, like, hey, Bill, it's your turn to go buy the pantyhose this week, and I was like, okay. You get used to it after a while. So I need you to know the overall architecture here. So I'm going to tell you in advance because we have this long break between the end of classes and when you actually write your exam. So I'm going to tell you in advance that parts of this slide are going to be on your final exam as both a short answer question as well as several multiple choice test questions as well. And it may not seem like there's a lot here, but there is an awful lot that I need you to make sure that you are able to tell me. So when we are looking at sensory signals, I told you sensory signals come in, and I'm going to start at the bottom. Sensory signals come in to the entorhinal cortex. This is sitting off to the right. And these sensory signals from the entorhinal cortex, sight, sound, all that sensory signal, comes in. Those axons from the entorhinal cortex are known as the perforant path. So all of the axons together as a bundle, we would call that bundle of axons this perforant path. And these group of axons, shown here in green, will make their first synaptic connections to this area of the hippocampus, which is known as the dentate gyrus. So if you're going back, this is what I've been telling you about in this particular diagram, and you're just seeing it in a slightly different way. The dentate gyrus consists of a number of different cells. We've looked at the dentate gyrus. We've looked at granule cells within the dentate gyrus before, and we will see the first synaptic connection here at the level of the dentate gyrus as these granule cells. So this is one synapse that you are seeing here. These granule cells in turn will send their axons to the next group of cells within CA3 of the hippocampus. All of their axons collectively are known as mossy fibers. So I could, for example, stick an electrode here and stimulate the axons of the mossy fiber pathway. I could stick an electrode out here and stimulate the axons of the perforant pathway, and I would observe the responses on the granule cells. That's usually how I ask these questions to make sure that you understand the anatomy of the hippocampus. So in turn, these CA3 pyramidal cells will send their axons, collectively known as the Schaffer collaterals, to the CA1 hippocampal neurons. This pathway is the most studied pathway in all of neuroscience. It is impossible for you to get through a neuroscience program without knowing or hearing about this pathway where you have these CA3 axons called the Schaffer collaterals, which are involved in LTP. We stimulate these Schaffer collaterals. We cause them to fire action potentials, and then we see if these neurons have stronger synapses at the end of that stimulation. A couple of other things that I'll just make note of. If you were to look across the entire brain, everywhere in the brain, If we're to take a look across the entire brain, we would see that the vast majority of LTP, strengthening of synapses or long-term potentiation of synapses, involves NMDA receptors. We need NMDA receptors to cause LTP, except for one place. And there are very few places in the brain where NMDA receptors are not involved. We can strengthen this synaptic connection between the MOSI fibers and CA3. We can strengthen those, but we don't need NMDA receptors to do that. We don't have to activate NMDA receptors to make this synapse stronger. It is not NMDA receptor dependent. It is dependent in CA1, so this is NMDA receptor dependent. These granule cells in the perforin pathway, NMDA receptor dependent. Your cortex, NMDA receptor dependent. The nucleus accumbens, NMDA receptor dependent, but nowhere else. Like here in the CA3, we don't need NMDA receptors at all. And that is a fact that we'll come back to again, but I do want you to make sure that you are learning that now. Okay, so again, this is just a different view of the same thing. And I want you to think very, very carefully about this. And you don't need to memorize all of this. It's just a different way to see the exact same diagrams that I've been showing you in different ways. You should be able to understand all of this. You don't really need to know CA2 here, but we're going to come back and look at this overall circuit later on. And this circuit that Candel is always looking for, this circuit is important for us as modern neurobiologists. So I want you to think a little bit about this, and I think we'll wrap it up a little bit early today. Do you think that every single synapse is plastic all the time? How many of you would say yes? Synapses are always plastic. Doesn't matter at any given point in time, all synapses are plastic. Yes? How many of you believe that the brain is inherently got plasticity? Yeah, so you agree on that? I would agree with you. But why then can't all synapses be plastic all the time? Yeah, Stefan?
Synapses are too plastic. Wouldn't you wake up and have to relearn how to do everything?
Right. So there's this really fundamental concept that we can't have too much plasticity. We can't really have plasticity to a level where if they're always changing, that we might actually be losing memories as we go along. And this sets up what we're going to be talking about in the next lecture. And this is the biggest controversy that involves research labs here in Toronto, by the way, where we're looking at, are those younger synapses, are they actually much stronger and are they much more plastic than these older synapses that many of you have? And if that's true, why are those synapses actually involved in amnesia versus our synapses, which we think are involved in learning as well as in memory? So we're going to be looking at how we have this synaptic plasticity and why we don't always have synaptic plasticity and why that's actually a good thing for our brains not to have too much synaptic plasticity in the next lecture after break. So a couple of you were asking during the break, and I'll just take 2 minutes of your time and we will end early. I am intending to go back and make those weekly study guides and some practice questions for you. I've just been a little bit behind schedule and I've been a little bit tired, to be honest. I know many of you are as well, so just bear with me. I will catch up, just like you'll catch up during your fall break. So just give me a little bit of time. I'll make sure that you're well prepared for the final exam, so not to worry there. Number 2, please don't leave your literature review to the last minute. So many of you are looking at, wow, I have a whole month to be able to work on this literature review. So number 1, if you are going to change your paper, go ahead and do so, but come and talk to me before you do that. And number 2, you'll see the rubric sort of out there. So there's a list of things that you should be doing in your literature review and a whole list of things that you should not be doing. And we want you to have more on the do side than on the don't, and that's how the TAs are going to be evaluating you when they're looking through your rubric, they're looking through your literature review. You are, by the way, always welcome to send an early a version of your lit review either to myself or to the TAs. You'll know which TA is going to be responsible for your lit review. You'll get your grades up. You'll see who marked your visual assignment, so you can always communicate with them. They're always willing to help you with that, or come and see me and I'll help you with that as well. Do you have any other last minute questions outside of what I've just covered? So I'm going to wish you all a really restful fall break. Please take some time off, get some rest, enjoy yourselves a little bit, and come back the week after ready to get going on the second half of the semester.